Hey guys, welcome back to my channel. This is today's second video. So today we are gonna start with biology's first chapter, which is cell cycle, chromosome, and genetics. Will be done in the next video, that is tomorrow. So now I'm gonna start with cell cycle and chromosomes. So let's begin. So first of all, what is cell cycle? See, if you take a normal ordinary human cell, this cell undergoes several processes to give or to produce daughter cells, and the processes which it undergoes is known as the cell cycle or the see if you take a cycle, you pedal it continuously in the same way, rotation, and here also there's rotation of cells. You all started as a single cell, even me too, and now we are a organisms with millions of cells. Okay, so first of all, the cell cycle is a sequence of events for cell growth, DNA replication, and division of cells. Okay, so it ensures proper functioning and reproduction of cells. Dividing the cell cycle, we get there are two phases in the cell cycle: interphase, mitotic phase. So first we'll go with the interphase. I think interface is very easy, so I'll explain it shortly, but mitotic phase I'll explain in detail. So I'm gonna post this in the PDF form in my Telegram channel, which I've started today. The name of the channel is Amog's ICSE Notes. So you can take up this there and you can study from it. I'm gonna even add diagrams from next video so that it's more interactive to you guys. So let's get ahead and start this. So first of all, G1 phase. So what is G1 phase? Growth 1 phase or first growth phase is known as G1 phase. In this phase, the cell grows, performs its natural function. See, now a cell has exited mitotic phase and it started again growing for the next cell division. See, I took a single parent cell. It divided into two daughter cells, and those two daughter cells are yet small, but once they enter the interphase, they grow in the G1 phase. Mainly they grow and perform normal function like absorption of oxygen and production of ATP and storing of ATP in mitochondria. I think all you all know water cell processes. And next, once they grow to a full size like the parent cell, the daughter cells enter mitotic phase. So first in G1 phase, those cells grow, the daughter cells grow into to reproduce or get bigger, and these daughter cells synthesize proteins and duplicate their organelles. See, each cell has a set of organelles: nucleus, mitochondria, centrioles, lysosomes, endoplasmic reticulum, rough endoplasmic reticulum, smooth endoplasmic reticulum, nuclear membrane, etc. etc. And example, see, liver cells repair tissue during G1 phase. See, if there is any damage in liver, that is repaired especially during G1 phase. Okay, when the cells grow, they repair the tissues. So next, S phase. In S phase, what you get is the DNA replication occurs, doubling genetic material. See, now think, for example, this is your DNA structure. Okay, 1, 2, 3, 4, 5, 6, 7, 8, 9. I'm gonna just draw in random. You guys. Okay, now what these are two strands of the DNA. I'll name it as A and I'll name it as B. Now what happens in S phase is these A and B grow to make C and D. So I'll explain it now. Wait. This A extends like this and forms a duplicate of itself. This A forms a duplicate of itself and this is named as A and C. So this single strand divides into two strands. This one strand of A divides into two strands and duplicates the DNA for the next daughter cell. And here even this is the same. Here also it happens the same. Here it's named as B and D. Guys, please don't mind my handwriting. I'm writing with a mouse. And if you guys support me by subscribing me and liking me, I'll change into a tab pen, or a pen tab, sorry, my bad, and my handwriting will change. I'll write with a pen neatly, and I hope you guys understand then. So please like, share and subscribe, and spread me and my videos a lot. Send them to your friends, your family members, whoever are studying in the same class which you are, who are studying in ICSE. Send them, even send to CBSE students, as the topics are mostly related topics. Send them to everyone, let everyone learn from my channel. And I hope you guys support me. I hope you are watching my videos, because from the last 2 days, what videos I have uploaded, I got nearly 36 views on those videos. But I have seen that out of those 36 views, the subscriber count is not growing. I have only 2 subscribers right now. There was third person, I don't know who was it. He unsubbed me. But I hope you all support me and share my videos, whoever are watching. And I hope you all spread me, so that I can improve my handwriting by getting a pen tab or something which will help me write better and explain it to you better. Okay, next. I hope you all understood S phase. In this, the DNA is replicated, and some more. Each chromosome has two identical daughter cells. See, due to this, what happens is, think this is a single chromosome. Okay, this is a, okay, wait, my bad. Yeah, this is a single chromosome, guys. Okay, this is replicated during S phase. At they both, this is the original and this is duplicate. As DNA replicates, they both replicate, and a new one is formed here, which this point is called as centromere. Okay, yeah. So next example, human cells replicate DNA during S phase, increasing their DNA content from 2N to 4N, as I had showed you earlier. So this one, yeah, 2, and this divides into 2 more, so it forms 1 2 3 4. Okay, next G2 phase, second growth phase. Okay, further growth and preparation for mitosis. Here the preparation of the cell to enter mitotic phase begins. Okay, here the cell begins to prepare for the mitotic phase. Proteins which are required for the cell division are synthesized here. Skin cells in animals rapidly prepare for division during wound repair. So recently I had an accident, and during my wound repair I went through the G2 phase, or the or second growth phase. Okay, let's move ahead. See, first I'm gonna show you an overview which is of 2 points. I think I'll just read it out, you will understand. See, composed of mitosis, nuclear division and cytokinesis, which means cytoplasmic division. So there are two, in mitosis there are two processes, karyokinesis and cytokinesis. And this is an example of mitosis. You can read it. Stop the video, take a screenshot, I don't know, or when I'll send, read it then. So yeah guys, prophase. So in prophase what happens is, the centrioles start moving apart, and the chromos and reach the opposite poles. The centrioles reach these two poles, and next the chromosomes become distinct. Means the chromosomes are more visible, and the chromosomes are already duplicated. As I had said, in S phase the chromosomes are already duplicated as paired chromatids, as I had said you in S phase. And next, if we go ahead, the sister chromatids attach or get attached to the other at a small region called centromere. So I had said you earlier, what's centromere? A point where the sister chromatids are attached. And next, if we go ahead, spindle fibers appear between daughter centrioles, forming achromatic spindle. See, these spindle fibers appear here. The spindle fibers appear between the daughter centrioles, making um, achromatic spindle. Okay, Next, nuclear membrane and nucleus disappear. As I have written here, the nuclear envelope, which is the nuclear membrane, and the nucleolus are totally disappeared. And next, if we go ahead, we will go to metaphase. In metaphase, the chromosomes line up on the equatorial plate. Here the chromosomes line up, and see, each chromosome get attached to this. Here, from here spindle fibers are coming, you think, and each chromosome is attached to a spindle fiber from here. Next, anaphase. In anaphase, what happens is centromere attaching the two chromatids divides and splits. Here in anaphase, I have not given much here, but I know I'm telling. You can just write this down by pausing the video. So I'm telling it right now. As centromere attaching the two chromatids splits and divides, splits or divides this centromere. So this is a chromosome, you think, one, two, three, four. This centromere here splits the chromosome into two parts, one, two. Okay. Next, a furrow starts in the cell, and in the middle of the cell membrane, a furrow starts developing. So think this is the cell. A furrow starts here in the middle of the cell, developing a furrow. What's the furrow? A depression is known as a furrow. And here furrow starts developing. The two sister chromatids of each chromosome separate and move to the opposite poles. Okay. Next, telophase. What happens in telophase is the two sets of daughter chromosomes reach the opposite poles, which these were pulled by the chromatin like spindle fibers, right? They reach these poles, the two sets of chromosomes, and spindle fibers get disappeared, and next chromatids thin out in the form of chromatin fibers. Next, the nuclear membrane is formed. The cleavage furrow starts deepening in the animal cell. Nucleoli reappear. I think you all know what's nucleoli. So at last we go through cytokinesis or cytoplasmic division. See, a cleavage furrow. As I had shown you before, what's a cleavage furrow? Here there is a furrow forming. So what this furrow does is it deepens, and this furrow it deepens so much that the two new cells separate from each other and form, like the parent cell separates from each other, from the parent cell separates and forms two new daughter cells. Okay. Next, if we move ahead, we get meiosis. What's meiosis? See, in meiosis, first of all, I'm gonna explain it. Then I'm gonna tell what's like what I have written here. I'm gonna just first I'll explain what I know. Then I'll tell what I have written here. So chromosomes condense. Here six chromosomes have been. So wait, I will just take a circle, and here I'm gonna take six chromosomes. How many? Six. One, two, three, four, five, six. So here I have taken six chromosomes, and in which three are from male, three are from female. Okay, this is following 2n rule. But as we move ahead, okay, as we move ahead, this 2n rule, these six chromosomes get attached to each other and form three chromosomes. How many? Three chromosomes. Okay, they get attached to each other and form three chromosomes. And what happens to these three chromosomes is they get divided, but it's still in 2n, but these three get divided into n and n. So in n and n, what happens? I'm gonna tell you. So wait a minute, I forgot to draw. Here there's a count of total 8 single, like a single sister chromatid in one, 8 in each. So what happens here is 6 go here, and sorry, not 6, not 6, 8 0 24, like 12 go here and 12 go here. 12 12 one, like if you think this is a chromosome, right? in this one is a part, like this 12 go here and 12 go here, and from these 12, these 12 each is divided into two parts. From this 6 goes here and other 6 goes here, and from this 6 goes here and other 6 goes here. So here at beginning we had 24, that's divided by 4 and we have 6 each. Okay, now I think you all understood what's meiosis. It's a reductional division. It gives, if you take 24 chromosome, it gives you 4 chromosomes, means 4 gametes, 4 gamete cells. Okay, next it converts the 2n rule into n rule. And crossing over, I think most of you know what's crossing over. I'll just explain it again for guys who don't know. The process of exchange of genetic material during meiosis between two non-sister chromatids of a homologous pair. See, this is a homologous pair of chromosomes. Here a chiasma is formed where they both meet, and the part of this goes to this and the part of this comes here. Okay, this forms a total homologous chromosome, and crossing over happens here, due to which there's genetic variations, and this genetic variation causes changes in child's, and so the child's mostly, not mostly but nearly 40% differ from their parents. Okay, so that's it for today guys. Thank you for watching. Goodbye, hustle hard, happy learning, tata.